Hi, everyone. It's truly a pleasure being able to talk to you today about my work with the King Lab, investigating how helminths regulate the intestinal stem cell compartment. Helminths are really fascinating creatures. These are large microparasites or intestinal worms, as you can see from this image, that are mainly soil transmitted. They infect over a quarter of the world's population at any given time, mainly in sub-Saharan regions, as can be seen from this image. And importantly, there is currently no available vaccine, and these infections are also ubiquitous in livestock, making them of great importance in agriculture. From an immunological perspective, it's really fun to investigate helminths because they have really contributed fundamentally to a lot of what we know about immunology. This includes a lot of what we know about type 2 immunity specifically, but also the identification of new epithelial lineages and new immune cells. These types of infections also regulate diverse diseases in many different tissues, which makes the study of helminths really able to contribute to a lot of our understanding of basic biology and different disease settings. One might wonder, why are helminths such successful pathogens? And we can hypothesize that the fact that they're easily transmittable, that the host is not protected from reinfection, and that infections are highly chronic all contributes to the high pervasiveness of these pathogens. Which leads me to one of the main questions that we're interested in our lab, which is trying to understand why really are helminth infections chronic. And again, we can hypothesize that the fact that these parasites have been co-evolving along with their hosts for millions of years, along with the fact that they're highly immune suppressive, contributes to their ability to mount a chronic infection. In addition, these infections are also very well tolerated. And if you will look at this depiction of a 14th century French pilgrim infected with a guinea worm, you will see that while this guy has a very, very big worm creeping out of his leg, he's still standing up, looking relatively fine, right? And indeed, we know that while there are health costs stemming from these types of infections, mortality rate is extraordinarily low. This is also true, of course, for our mouse models of helminth infections. What I'm showing you here is the life cycle of our favorite worm in the lab, Haligmusimoides polygyrus baccari, or HPB in short. And as you can see, infection starts with the ingestion of infective larvae. They cross the epithelial barrier and embed themselves in the submucosa, where they form these very large granulomas that you can see here in this histological depiction. Later on, mature worms are breaching the epithelium once again, this time to establish themselves in the intestinal lumen, where they're wrapping themselves around the intestinal villi, and they will breed and they will lay eggs in order to restart their life cycle. And as I'm sure you can appreciate, there is a lot of potential tissue damage that can occur during the different stages of this infection. And when I first came into the King Lab, I was really fascinated by this aspect of tolerance that seemed to be manifesting during helminth infections. And I was specifically interested in addressing what's the interaction between the worms and the epithelium, not only because of the very close proximity between the two for many weeks and months at a time, but also because the epithelium is really at the nexus of host pathogen interactions in general and host helminth interactions specifically. We know that it not only poses as a physical barrier, but it's also the first to sense and initiate an immune response towards a pathogen, and it also actively participates in fighting the infection. What we do today is trying to have a very holistic view, working both in vivo and in vitro, assessing the status of infection, but also tissue integrity, what's going on with the immune response, both locally and systemically, and most relevant to my work, really investigating what's going on in the intestinal stem cell niche. And we're doing that using very diverse tools that I'm just highlighting here. When we first started this project, what we aim to do is to establish an organoid model to assess the intestinal stem cell response to infection. What we do is extract all of the different molecules that are secreted by these worms. This is known in the literature as helminth excitory secretory products, or HES in short. And when we first stimulated organoids with this HES, what we found was this very robust and distinct phenotype of large hollow spheres that took over our cultures. And to make a very long story short, what we later found was that HPV can actually directly reprogram intestinal stem cells into a fetal-like state. And this fetal-like state is also a manifestation of a broader pro-regenerative transcriptional program. We later found that HPV is also able to expand a very specific stem cell population known as revival stem cells that you can see here in red. And this was occurring simultaneously with the suppression of secretory cell lineages such as Paneth cells, goblet cells, and tuft cells. And we, of course, later on validated all of this in vivo, but I want to show you just one piece of data from those in vivo studies looking at revival stem cell lineage tracing experiments that established the fact that these stem cells are not only expanded during infection, but also that this specifically occurs during the luminal stage of infection, as you can see here at day 12. We later really wanted to assess what's the fate of revival stem cell daughter cells. And therefore, in these lineage tracing experiments, we also looked for the expression of tuft cells and goblet cells. And what we found comparing tomato positive, which are the revival stem cell progeny cells, with tomato negative villi, was the fact that revival stem cells actually give rise to less secretory cells. And when we stimulated organoids with IL-13, in the presence of helminth secretory products, we were able to see a complete suppression of secretory cell lineages, specifically tuft cells and goblet cells here, that were suppressed with helminth secreted products. So to summarize this work, what we show is that helminths are secreting molecules that are able to specifically expand a revival stem cell population. We also show that this is mediated by an oxidative stress response in the intestinal crypt, and that these products are also able to suppress type 2 induced differentiation of secretory cells. Reciprocally, we also show that type 2 immune cytokines are able to suppress revival stem cell expansion, really indicating there is this type of tug of war between helminths and type 2 immunity in the control of the intestinal stem cell compartment. And our current working hypothesis is that helminths are specifically inducing this revival stem cell population that's been shown to be not only damage induced, but also critical for gut regeneration, in order to maintain really the barrier integrity. And what we think is that this might be a mechanism for the worms to maintain a chronic infection on the one hand, but still make it tolerable enough for the host. And we, of course, have very specific questions that we're currently working on, including identifying how does oxidative stress promote this fetal reversion, and whether or not the expansion of revival stem cells and secretory cell inhibition are two mutually exclusive events, or whether or not they're interconnected. And we're also trying to identify the molecules secreted by these worms that are responsible for this phenomenon. And of course, we have many long-term goals that I will be happy to discuss with anyone interested later on. And with that, I would just want to thank our little King Lab beehive and our queen bee, Ira, who has been extremely supportive throughout this project, and our very close collaborator, Dr. Alex Grigoriev, who has not only taught us basically everything we know about stem cells, but also shared with us a lot of the different tools that we're currently using. And thank you for your attention.